Welcome to this webinar on new perspectives on COVID-19. This webinar is being organized by Academia Europaea's Cardiff University Knowledge Hub and is being promoted by Academia Europaea and is also being promoted by the American Physiological Society's open access journal Function. And this is appropriate because several of today's speakers have recently published papers in Function that deal with COVID-19. Before we go on to the theme, I will just say a few words about Academia Europaea. It's as the name indicates, a European academy that elects members from the whole of Europe simply on the basis of scientific excellence. It covers all areas of science and the humanities. The most important external role of Academia Europaea is its participation in the European Commission's scientific advice mechanism, which provides independent science advice directly to European commissioners to inform their decision making. Together with 4 other pan-European academic networks, Academia Europaea produces evidence review reports in fields that are demanded from the European Commission. Academia Europaea does all its work for SAPEA, which stands for Science Advice for Policy by European Academies, from the Cardiff University Hub. So the Cardiff Hub is actually the key part of Academia Europaea that does the work for Academia Europaea for the science advice mechanism. It started at the end of 2016, got really going in 2017 with the first meeting of the SAPEA board, as you see there on the screen, with the group of chief scientific advisors. And as you can see from the picture, members of Academia Europaea play a key role in this advice mechanism, which is set to continue here in the new commission period. Now, my interest in this particular topic that we're dealing with today came about because a particular type of intracellular calcium signaling seems to have some interesting features in common with the coronavirus uptake. And to the right of the screen, you see a small sketch that illustrates some of the points that I found particularly interesting. So, there's no question that coronaviruses, and that includes SARS-CoV-2, are taken up into the cells via the process of endocytosis. And in endocytosis, you move from early endosomes to late endosomes, and during that process, there is an increased acidification of the endosome. So as the pH falls, the calcium concentration falls. And that occurs through a 2-port channel, which is activated by an intracellular calcium-releasing messenger, nicotinic acid adenine dinucleotide phosphate, abbreviated here as NAADP. And what interested me in this reading the early literature on SARS-CoV-2 uptake was that the uptake of that virus is totally dependent on this endosomal acidification. If you block the proton pump with bifidobacterium, for example, there is no SARS-CoV-2 uptake at all. It's gone. Similarly, if you inhibit the 2-port channel that is activated by NAADP, the virus uptake is also dramatically reduced. So I think this gives some interesting potential opportunities in the future for therapeutic interactions. And I wrote this article in Function, which is posted on the left of the screen. So that's all I have time for in terms of scientific presentation. And I will just introduce the speakers now. So we have 5 outstanding speakers, all members of Academia Europaea, who will talk to us today, starting with Valerie O'Donnell, then follows Giuseppe Novelli, and then Jose Lopez Barneo, followed by Bernard Rossier, and finally, last but not least, Alexei Verkratsky. And in order to keep the time into the 5 minutes that I allocate myself, I will now, without any further ado, give the floor to Valerie O'Donnell from Cardiff University School of Medicine, who will talk on targeting the viral lipid envelope of SARS-CoV-2. So, well, the floor is yours.
Yeah, just sharing my screen. And can everybody see that?
Yeah.
You can see the presentation. Yeah. It should come up as a presentation as well. Okay, so thank you very much for the opportunity to present on this, Ole. It's been a really interesting journey so far. And I think we're getting to the point now where we're going to hopefully get some answers to this soon. But how I got into this, how I started asking this question about oral rinsing and mouthwash was that I'm a lipid biochemist. So I'm interested in lipids. I work a lot on lipid membranes in mammalian cells. But early on in the pandemic, we were hearing very strong messages from the World Health Organization, the NHS, UNICEF, and all of these agencies on how important it was to wash your hands and use alcohol-based hand gels and soap. And the reason for this was widely stated that it was because this virus has got lipids in a membrane, and so you're dissolving the lipids. So I got quite intrigued by this, being a lipid biochemist, and I decided to look into this. And I think the first thing that I heard from speaking to microbicide experts actually was that, you know, most microbicide approaches are broad spectrum. They're there to inactivate bacteria and all sorts of viruses, but that this virus and coronaviruses in general, all of them plus influenza virus and many other viruses we know of, are actually what is termed enveloped viruses. These viruses are far more sensitive to chemical inactivation than bacteria or non-enveloped viruses, and thus a virus like this should be quite easy to inactivate using chemical means. And I found that really interesting. I thought, well, let's look into this a bit more and see whether the approach we're taking on our hands could be adapted to, the site where the most important viral replication is probably taking place in early illness, and that's in the throat, where we swab for the virus, where we find it and where we test for it. Clearly oral virus appears to be a source of virus that gets transmitted to infect disease. So I brought together, I'm a lipid biochemist, I don't really have expertise in virology or microbicides or other areas of relevance to this, the immunology side of things, dental hygiene, etc. So I brought together a mixture of people, some of whom I'd never met before, and interviewed them and talked to them and looked in the literature and wrote this review back in March, April time. And what we concluded was that really there are many existing mouthwash formulations on the shelf that are known to have the clinical ability to reduce inflammation in the mouth. You know, they're there to prevent gum disease. And in theory, based on how we know these compounds interact with lipids and lipid membranes, they should have efficacy against this virus. So we wrote this review, and a really sad thing happened was that Michael Wakelam, who's the picture on the right, who's one of my co-authors, he actually passed away due to COVID in the middle of the time we were writing this review, which kind of made me even more determined to push it through and follow through on this. We had a lot of help from Boots and Johnson & Johnson, who gave us access to formulation information that we couldn't have otherwise got, so I do want to credit them. And you can see here in this electron micrograph, it's going to be here, this green part, this is the lipid membrane. You know, we got a lot of research on the proteins, but relatively little in terms of the lipids, and this is where we really need to go with this. So what our question really was about was this part here, this membrane. You know, can it be targeted by these lipid disrupting compounds, which are in commonly available, non-toxic, cheap mouthwashes that you can buy in the pharmacy, known to have efficacy against microbes and inflammation in general. And we looked into this and concluded that yes, there is clear evidence that they work against some other enveloped viruses and that they really should be tested here, but that really there's no gold standard clinical trials done on this. And actually, it's virtually impossible to do double-blind randomized clinical trials on something like this. It's very much like the face mask story, where it's a real challenge. And in certain countries, particularly in Asia, where they've dealt with SARS and other respiratory viruses a lot more than we have, they have done some trials, but they're not of the standard that WHO and public health bodies would consider good enough to allow recommendations to be made now. So what we know about this virus, or coronavirus, is that the lipids bud from ER Golgi intermediate complex. They're going to be comprised of generic host-derived lipids, but we need to measure this, but we're doing that now. We predict they're not going to change due to mutations in the viral genome. I think that's a really important point. So if you find something that targets them, it should continue to work. And it's accessible, and shed virus is part of the transmission of this virus. But not all viruses shed. A lot of it's going to be inside cells. If you rinse it out, there'll be a continuous replication cycle. So we really need to know if there is an impact, how long does it last for? And so this was where we were. So what's happened since? Since the review came out, there was a lot of media interest. It's already been featured on something like 50 media sites, and still going. We've got another interviews going on this week that will be broadcast on the radio next week on this area. And I think it's taken time, but it is starting to achieve more interest more broadly now, particularly in the public health arena. There's been 5 preprints and published papers, which are listed here, all looking in vitro, and which have been really positive, I would say, including one from Dave Thomas and Richard Stanton, who are at Cardiff University. Some of these are from the States, from Germany, from all over the world. They've all taken an in vitro approach where they have taken either the current SARS-CoV-2 virus or a very similar strain. They've incubated it with a soil load, which mimics mucus in the back of the throat, and then given it a 30-second incubation with mouthwash, various mouthwashes, rinsed away the mouthwash, and then put the virus onto cells in culture to see if it's still infective. So it's an infectivity assay, which is very important, not PCR. And in all cases, we find really strong efficacy of mouthwash, various formulations. And I think one that I will show a little bit on here is this is work from the preprint from Cardiff. Rich Stanton set up this assay so that he was able to test activity to achieve an international standard in order to be able to call a formulation a viricide or not. And at least 2, this is CPC, cetylpyridinium chloride, and then this is ethanol plus surfactant called ethyl lauryl arginate. These preparations completely inactivate virus in this assay, meaning that they can be called officially viricides to an international standard. So in vitro studies have been really positive, really clear, I think, that it can work. But what about in vivo? I think the real thing that would impact on public health advice is knowing whether this works in vivo. So far, it's still very early days. This review that was published early this year has listed a whole load of clinical trials on clinicaltrials.org, which are ongoing now. These will all be in patient groups looking at I assume this is going to be efficacy in terms of inactivating virus. It's very difficult to test transmission. You need huge population groups or patient groups for that. So looking at inactivation in the throat is the first place to start, and it'll be really interesting to see what they report. There have been 2 published in a preprint, but unfortunately these are very small, and you can't draw conclusions from studies like this. I would say they're anecdotal at best right now. But if you look at this one on the left, there's 4 patients with detectable virus basally. This is by PCR. And when they were rinsed with an iodine mouthwash, the 2 patients with the higher levels, it came down and it stayed down at least to 3 hours post-rinse. The other 2 have got low virus basally, so you can't really draw any conclusions. But as I said, I think this is too small to really make conclusions about the clinical ability of these agents to be able to work in the back of the throat, and we need more data. So we in Cardiff are doing some work on this. Rich and Dave are continuing with a clinical study, which is similar to the others that are published, but hopefully with a larger numbers, which is what we really need. Rich is testing a number of different compounds against the membrane to look for a lack of infectivity. We're also doing lipidomics, mass spectrometry analysis of the membrane of the virus. And the last thing I'll say is WHO, having said for months that there was no evidence to recommend this, now, since August last year, have been recommending that dentists do include, for the purpose of reducing the load of microbes, including this one, that a mouthwash is used. So I'm very, very glad to see that. And I'll finish there. Thank you, Ole. I've run out of time.
Okay, thank you very much, Val, for this very interesting presentation. We will have the discussion at the end of all the presentations. And to those who are thinking about making questions now, I would just advise you to formulate them very concisely. Sometimes at these webinars, we get very, very long questions, difficult to deal with. So make brief questions that are precise and clear. So now it's my pleasure to call on Professor Giuseppe Novelli from University of Rome, Tor Vergata, to give his talk on genetics underlying severe COVID-19. So Giuseppe, the floor is yours.
The slide, that's okay?
Yes.
Yes, okay, thank you. Thank you all for the opportunity to appear today, and thanks for the invitation to this important webinar. Okay, as stated by Tony Fauci, as a very famous virologist in the US, understanding a disease that is both asymptomatic and later cases is a major challenge at the moment. During the last year and this year, we just studied the possibility that we have different groups of the people affected by COVID-19. We have asymptomatic patients, we have patients with mild or moderate symptoms, patients affected by severe symptoms that require hospitalization, and people with critical symptoms that require ICU at the hospital. And this is very difficult and very dangerous for the life. Symptoms of COVID-19 include symptoms most predictive in terms of SARS-CoV-2 infection are hereditable. This is what this means, that about many of the variants observed in the phenotype of these patients is due to genetic factors. Some colleagues from London last year estimate that hereditability of COVID-19 is included in a percentage of 30% to 70% of the factor influencing this important disease. As we know, it's many other infectious diseases are influenced by genes of the host. And to this scope in discovery genes and alleles involved in susceptibility or resistance to disease, many groups have been organized very important consortia. Just here you have a list of the most important genetic consortia to study the genetics of the host involved in COVID-19. One of these important consortium is the COVID Human Genetic Effort, is an international consortium coordinated by Jean-Laurent Casanova at Rockefeller University in New York. And in this, the aim of many of these consortia is to study and to stratify in order to distinguish people with major risk or minor risk to the infection disorders, because we have the same virus, but people are different. In terms of, you have people show with some genes or alleles, if you like, in terms of resistance to infection or more susceptibility to manifest a severe disease in young people, for example, or others. And in using the classical genetics approach, using, for example, wide genome scanning or candidate gene analysis in the patients of different groups of phenotype, starting from asymptomatic or severe or moderate or resistance people, many people established that the studies that the individual many alleles, many genes involved in this susceptibility. And in terms of population genetics and epidemiological studies, most association studies have been performed and identified many alleles that confer a specific risk to develop this some or specific form of the diseases according to the genotype. And just here, I would like to show you many of these genes identified. Many of these have been confirmed in different labs. This has come from my recent review now submitted in Human Genomics, in which we performed a very careful analysis in order to establish which genes and which alleles are involved in this, in order to establish risk estimation in these disorders. I just would like to separate in common variants like HLA or HLA-DRB1 or ApoE, HLA-G, and other genes involved as TMPRSS2 is involved in the entry of the virus in the cells, but also other genes involved in other physiological aspects of the infection. Together with the common variants in which we have very high frequency in the population, like blood group, for example, A or 0, are very, very frequent in the population, but we also were able to identify genes with the rare variants that can confer a high risk to develop a specific phenotype of the disorders. As you can see here, some genes are reported here, or some allele, but what is important, we and other people identify the ultra rare variants in a set of particular genes, and in particular, genes involved in the interferon pathway. As you can see here, you have TLR3 or IRF3, IRF7, all these genes coding for protein involved in the interferon pathway. And we and others were able to identify this ultra rare mutation in a very large group of severely affected patients. And you can see many of these are deceased, and we are this list, the genes, the mutation, the age of the patients, and the ancestry of the ethnicity of the patient in which we identified these particular genes or mutation. What is the importance of this paper? This paper is published on Science in October, and you can see here we compared asymptomatic group of people with very, very severe group of patients. And in 3% of this group of patients, we were able to identify specific mutation in recessive or dominant model in genes involved in the interferon response to the infection. And this was the first demonstration that genetics of the host is very, very important in a subgroup of patients affected by COVID-19. But in an additional group of patients we studied, in about 10%, we were able to identify the presence of autoantibody to type 1 interferon. Therefore, no mutation in genes, but the presence of autoantibodies to IFN. And this was important because the result is the same. You have patients that are not able to produce interferon, or patients that are able to produce, but is inactivated by the same people, by the same situation in terms to produce autoantibodies against interferon produced by self.
I'm sorry, Giuseppe, you have to conclude now.
Yes. Just a free slide. Yes, thank you. How we can use this information in terms for therapy for the future? As you can see here, one is to use interferon or similar to study, or interferon to give a patients in which you have a defective models. And this could be a personal approach. Another aspect, and this is very recently, we were able to identify mutation in a subset of genes involved in ubiquitin E3 ligases. And this is a very important ultra rare disease, but for these genes, we were able to identify some specific drugs that can inhibit the activity of these genes. These genes are important for the exit of the virus from the cells. And this, therefore, is important because these genes and this protein, of course, are important in ubiquitination of the spike protein of the virus. Therefore, we can use this information to stop, to block the activity of these enzymes in the cells. And this could be open the possibility to start with a clinical trial of this innovative drug. Just my last slide. I think that the genetics is important towards the personalized medicine for the future, as demonstrated for the HIV or for the AIDS diseases. Thank you very much.
Thank you very much. We had to move on swiftly to the next talk by Professor Jose Lopez Barneo from the University of Seville. And Jose will talk about silent hypoxemia in COVID-19. So the floor is yours, Jose.
Okay, thank you, Ole, for your invitation. I will talk on this silent hypoxemia in COVID. A very frequent complication in COVID patients in pneumonia. You can see an example here in this X-ray image, affecting mainly the lower part of the lungs. And as a consequence of pneumonia, there is an alteration in gas exchange between the alveoli and the blood. And therefore, there is a decrease in oxygen tension in the blood, what is called hypoxemia. In some patients, this decrease is very frank, very clear. PO2 oxygen tension goes below even 15 meters of mercury. And this is a very dangerous situation from the medical viewpoint because it may cause damage to brain and other organs. Normally, our body reacts to hypoxemia with what is called the chemoreflex, okay? And it's increasing the frequency of breathing, trying to incorporate more oxygen into our body. And we have the feeling of the lack of oxygen, of hunger of oxygen, what in medical terms is called dyspnea, okay? But some people presenting silent hypoxemia do not have this kind of reflex, they're not aware of hypoxemia, they're not breathing, or they're not dynamic, breathing at a higher frequency, they're not dynamic, and therefore they develop silent hypoxemia. And of course, this condition of silent hypoxemia make the situation even worse, okay? This has been a situation, the pathogenesis of this silent hypoxemia is a mystery, has been a matter of discussion by physicians and scientists all over the world. And we focus on one particular aspect because this chemoreflex, which is a compensated reaction of the body for hypoxemia, is normally mediated by a structure that we have been working on that already for many years, which are the carotid bodies. The carotid bodies are bilateral organs located in the bifurcation of the carotid artery. You can see that. These are their tiny glands which contain cells which are called chemoreceptor cells, which are able to sense oxygen levels in the arterial blood. And in conditions of a decreasing oxygen levels, these cells generate a signal which produces action potentials in nerves connecting this carotid body with the respiratory center to produce this hyperventilation, this increase in breathing that I mentioned before, okay? Here you can see a typical chemoreflex from a human being where you may are hearing the ordinate hyperventilation in the assist oxygen tension in the blood. And as you can see, as the oxygen tension goes down, there's an increase in ventilation that is very frank, very much activated at oxygen tension below 70-60 millimeters of mercury. This response, this chemoreflex that I am commenting, is completely gone in patients without carotid bodies, because in some cohort of patients that the carotid bodies were removed because of tumors in the neck, because surgery, etc., this patient lost completely awareness of hypoxia. So therefore, we hypothesize that probably the carotid body affectation, infection by the virus, by the coronavirus, is a contributor to the silent hypoxemia mentioned before. This idea, this hypothesis has been fundamented in two main points. One point that you can see here is related with the mechanism of coronavirus infection of cells. It is very well demonstrated that these viruses infect the cells binding to receptors. They are the angiotensin converting enzyme, ACE2, the type 2, and binding to these receptors is necessary for endocytosis and to infect the cells. So we have shown, we can look at whether or not this receptor is expressed in the carotid body. And here you can see immunohistochemical analysis of the human carotid body. This is a section of the human carotid body where you can see the expression in the red color on this receptor, not only the blood vessel, this is faint expression, which was known before, but also many of the parenchymal cells, the chemoreceptor cells, express high levels of this receptor. In addition, we have proven that this receptor is expressed in the carotid body, looking by quantitative PCR in the carotid body and showing that many male and female people, they are donors that we have obtained from our hospital, they express high levels, some of them express high levels of this ACE2 receptor, and therefore supporting the concept that probably at the beginning of the coronavirus infection, carotid body cells may be affected by the virus. And the second point I wanted to discuss, and the second evidence supporting our hypothesis, is related with the way what happens when one cell is infected by the virus. When one human cell is infected by the virus, the virus will hijack the protein synthesizing machinery of the cells, and therefore the host cells start making protein for the virus. And this produces a huge alteration in protein homeostasis in the cells, affecting mailing to mitochondrial proteins. It has been shown by several papers published in the last few months that mitochondrial proteins are very much altered in coronavirus infected cells. And this is very important because in our group, we have been showing in the last few years that the mechanism of oxygen sensing by carotid body cells depends on mitochondria. These cells have the mitochondrial-based oxygen sensor. They have a very active electron transport chain. They use oxygen as a final acceptor of electrons to produce water and ATP. But in response to hypoxia, in these chemoreceptor cells produce these signaling molecules, NADH and reactive oxygen species. These signaling molecules will modulate the potassium channels in the membrane. Here you can see in these microdomains. And inhibition of the potassium conductance will produce cell depolarization, opening of calcium channel, calcium influx, and secretion. Therefore, a proper mitochondrial function is absolutely required for oxygen sensing by carotid body cells. And therefore, we have proposed in this last slide, our model is that the carotid body chemoreceptor or glomus cells, they express these ACE2 high levels of these ACE2 receptors. They are infected by the virus. In the early phase of the infection, the virus may alter the mitochondrial sensor because I mentioned before this hijacking of the protein synthesis in the cell, and therefore the alteration of the mitochondrial sensor will make the people unable to detect hypoxemia. But as the infection progresses, there may be an immune response, inflammation, etc., and they even produce cell death, and therefore worsening the lack of awareness of hypoxia characteristic of these patients. We are now working on the carotid body from people who die from COVID-19 in our hospital. We are trying to confirm that the carotid body from these people are affected, are altered, and also we plan to do in the future. We want to see whether these affectations are just reversible or they are maintained for a long period of time after recovery from the disease. Okay, so in conclusion, our hypothesis is that the modification, infection of carotid body chemoreceptor cells may contribute to silent hypoxemia in these COVID-19 patients. And thank you very much for your attention.
Thank you very much, Jose, for this extremely exciting talk. It was very nice. So we will move on now to Professor Bernard Rocher from University of Lausanne, and he will talk about the critical role of sodium transport in airways in COVID-19. So Bernard, the floor is yours.
Thank you, Ole. Okay, so thank you for giving me the opportunity to talk about the role of ENaC in the infection of the airways. We propose in this paper a pathophysiological model for the early phase of airways infection. This model is compatible with the somewhat unusual progression of the disease. As you have seen before, this could be fast, slow, or silent, allowing transmission from asymptomatic patients. We will also show that the model is compatible with infection of alveolar type 2 cells, which is responsible for the early phase of viral pneumonia. And finally, it's also consistent with the pathophysiological model proposed by Garvin et al., so-called bradykinin storm, to explain the late phase of the infection. Now, as a background, we started from two publications. One from in silico study published by Annan and Hall in eLife, which showed that SARS-CoV-2 has acquired, unlike other coronavirus, has acquired a new sequence, 8 amino acid sequence, which is identical to a cleavage furin sequence found in ENAC. It was the first paper that attracted our attention. The second one is a nice methodological paper showing by the genetic methods that there was a variable infection gradients in the respiratory tract. This means a parallel expression of the ACE2 receptor for the spike protein and infectivity. So the infectivity was very high in upper airways and very low in alveoli. Surprisingly, and we'll discuss this later, club cells in bronchioles, which are critical for mucociliary clearance, were not infected by the virus. So starting from that, we asked 2 questions. What could be the role of MCC, mucociliary clearance, and specifically ENAC in the progression of the infection? And the second question, what would be the role of ENAC-mediated alveolar fluid reabsorption by pneumocyte type 2 upon alveolar viral infection? So the main entry of the virus is obviously in tissue where you have the highest expression of the receptor, and of course where the virus load is high. And this is obviously in the nasal, in the nose and the olfactory epithelium, and also in the mouth, oropharynx, including taste buds. So these epithelia express very high density of the endogenous COVID receptor and its co-receptor TMPRSS2, and they are easily infected by the virus. So the nasal mucociliary clearance will push the virus from nose to the pharynx, where the infection will progress down to the pharynx and then either towards the intestine and or to the lower airways. Mucociliary clearance in trachea and bronchi are extremely efficient in removing inhaled particles of pathogens and virus. And this is critically dependent on the height of the periciliar fluid, very thin layer, around 7 micrometers, requiring a perfect balance between chloride secretion, which is mediated by CFTR, the cystic fibrosis gene, and sodium absorbent mediated by ENAC expressed in ciliated cells. So ciliated cells are infected by the virus, and it is expected that MCC will be slowed down, allowing the progression of the infection down to the bronchioles. The bronchioles, we have a rather unique situation. The histology shows the simple columnar epithelial cells ciliated. The goblet cells, which secrete mucus, are progressively replaced by these club cells and neuroendocrine cells. And again, it requires a perfect balance between chloride secretion, CFTR, and sodium absorption mediated by ENAC in club cells. The importance of club cells is demonstrated by genetic disease like cystic fibrosis or PHA1 type syndrome, in which mucociliary clearance is strongly affected downward or upward. So since club cells are not infected, MCC should be less affected and progression of the infection towards the alveoli slowed down. This could explain, this strange progression of the disease. Now, when the virus reaches the alveoli, it has a very important effect. Indeed, in the alveoli, you find AT2 cells, alveolar type 2 cells, which express not only surfactant, which is well known, but ENAC, and this is critical for reabsorption of the fluid, which is secreted from the capillaries through the AT1 cells, the pneumocyte lining the alveolus. So when AT2 cells are infected, and this has been very well demonstrated in severely diseased patients, this leads to alveolar fluid accumulation, pulmonary edema, and a drop in PO2, which is very compatible with the early detection of so-called ground glass opacities, which we can see on CT scan, and which is, interesting enough, very similar to images of the wet lung syndrome of premature newborn, in which INAC is very poorly activated. So these are the model we propose. You find more details in this review, opinion paper we published in Function recently. And of course, I will be ready to answer questions later on.
Thank you very much, Bernard, for this interesting talk. And we now move on to the last talk in this series, which will be by Professor Alexei Verkrotsky from the University of Manchester. And Alex will talk about the psychiatric phase of COVID-19. If you could stop sharing your screen, Bernard, yes. Then Alex can get in.
I am trying. Do you see it?
Yes.
And do you hear me? Thank you very much, Ole, and good afternoon, everybody. And so today I would like to spend these 5 minutes to ask a very, very important question, because of course we all remember these horrible numbers for COVID-19, but at the very same time, our civilization now faces another epidemic, and that is the epidemic of neurodegenerative diseases. And here you can see what happens now, and you see that about up to 50% of older people, they do develop dementia. And that is something which probably reflects not only our genetics, not only our intrinsic things, but also environment. Because if we start to look what happened with dementia over the last 130 years, we see that these 130 years were accompanied with an increase in the risk of developing of this senile stuff. So these are the results which are taken from the book which was published in 1889, which is called Old Age, which looked into different organs and the systems in order to see what's going on with them in aging. And it turns out that the brain keeps aging better than any other organ. And here you can see numbers. So memory was good in 78% of older people who were older than 80 years old. And there were problems with memory only in 2 out of 74 centenarians. So now, of course, the situation is more dramatic, and most of the centenarians do show signs of senile dementia. Of course, it's very, very difficult to interpret, but nonetheless, it shows that probably environmental factors are having something to do with that particular process. And of course, infectious diseases and pandemics of infectious diseases could be one of those environmental factors. And indeed, over years and years and years, there were brain sequelae of infections. And one of those infections is very, very known thing, which is called encephalitis lethargica. And the last pandemic of encephalitis lethargica started in 1915. We still don't know what was the actual thing which caused it, and was it the virus or was it not? But nonetheless, if we start to look through the history, we see that these epidemics of encephalitis lethargica under different names like febris comatosa or Schlafkrankheit or coma somnolentum have happened in the world many, many times, and quite often they were associated with influenza epidemic. So now let us try to look what happens during COVID, what happens during this disease, which may affect the brain, and whether these things which affect the brain can have long-lasting consequences. And of course, the very first thing and the leading pathology which we are experiencing in COVID-19, as already Pepe Lopez Berneo has presented, is of course ischemia, because the leading thing in the pathology is respiratory failure associated with pneumonia. And what is ischemia? Ischemia is an energy, and brain is the most energy hungry organ in our body. About 20% of all energy is expended in the brain. And what is the major major molecule which takes all this energy is, of course, sodium-potassium pump, which restores ionic gradients. And those ionic gradients are of a paramount importance for information processing, information distribution, and everything which happens in the brain, from action potential to synaptic transmission. And of course, at the moment when oxygen saturation goes down by 10, 15, 20%, it already has a detrimental effect on the brain. Therefore, very many COVID-19 patients, especially at severe stages, they do present brain symptoms. They do present a number of neurological symptoms which are probably caused by this hypoxia. The second thing is, of course, systemic inflammation, and systemic inflammation accompanies COVID-19, and we learn that this is one of the leading pathogenetic presentations of this disease. And systemic inflammation is associated with a cytokine storm, with appearance of a very, very high concentration of pro-inflammatory cytokines in the circulation. And what it does, it affects blood-brain barrier. So it interacts with cells of blood-brain barrier, it leads to its destruction, and it leads to infiltration of the brain by different immune factors, which of course may cause autoimmune inflammation. And at the same time, it compromises the defensive function of the barrier. And that, of course, underlying a direct infection of neural cells, which can be through several pathways. And one of these pathways is indeed through fornical organs, where blood-brain barrier is non-existing or is much weaker. Another one is through olfactory system, because virus can infect olfactory neurons and progress through their process into the olfactory bulb and then to the rest of the brain. But nonetheless, whether this direct viral infection is very, very prominent, we don't know. At least there is not that many cases of true viral encephalitis. Now is thrombosis, and we know that in COVID-19, the whole thrombotic system is compromised, and therefore thrombosis are arising, and that causes stroke with obvious consequences. And of course, thromboinflammation, and again, epithelial damage, and BBB compromise, and vicious circle. And finally, all this COVID is associated with a huge psychological stress, because its disease burden is time in intensive care unit, is social distancing, is lockdown, is everything. So if you look into this scheme, you will see that all these factors together do affect the brain, and all these factors together may account for increase in burden of neurodegeneration, because we know that all neurodegenerative diseases, they are not purely brain diseases, they are systemic diseases. And whenever the system, the organism is ill, that causes an increase in neurodegeneration and in risk of it. So we can anticipate increase in that burden after the end of this pandemic. Thank you very much.
Okay, thank you very much, Alex, and thank you for keeping so well to time. That's very helpful. So we can now move into the discussion period. Some questions have come in, and I'll try to distribute them a little bit so that we have a varied discussion. And I also am very happy if other panelists will move in and make comments to some of the questions. So the first question is a kind of very general popular one, and probably one that Val would expect. But nevertheless, I think let's start with that, because it's probably a relatively easy one. But John and Angela Croat ask you, Val, whether in view of what you said, was President Trump at least partially right?
No, I mean, I think, yeah, this is an interesting one. No, Donald Trump said, inject yourself with bleach. And that is absolutely not what we're talking about here. We're not talking about injecting, and we're not talking about using bleach either. And I absolutely would not recommend that. I think it is important to point out, though, with any of these kinds of oral rinses, they should always be used in line with the manufacturer's instructions, which are very clear, that you rinse and then you spit out and you do not swallow. So it absolutely isn't the same thing. And I think it's a real shame when these things somehow get conflated, because it's clearly not the same. No.
I knew you would say that, but I still thought that we should just sort of.
Yeah, no, I'm glad you brought that up, actually. Yeah.
A more interesting question, perhaps, which I'm sure you may also have thought about, a question posed by Alison Fianta. She makes the point that according to her perception, in the Far East, people are generally using mouthwashes much more than we do in the so-called Western world. And could that be part of an explanation why these nations have done so very much better than the US and Europe?
Yeah, it's a very interesting question. It's very clear. I mean, in Japan, particularly, the death rates have been unbelievable in comparison to other Western countries. They have used them a lot more. In fact, they're part of their pandemic preparedness policy in Japan, even mentioned preventative uses of gargling. However, I think it's a question that isn't answered. There's other big differences. For example, the rate of obesity in Japan is only 3%, which is incredibly low. And there's other demographic differences and social differences that will be also undoubtedly a big part of why it's different. But I don't know. I think more epidemiology, more research needs to come out to really address this question.
Okay, thank you very much. And then there are several questions to Jose Lopez Barneo. So one person asks whether this DNA damage to the mitochondria, which could result in an increasingly heterogeneous mitochondrial population, could that perhaps explain the so-called long-term COVID?
Well, as far as I know, I have not seen any report describing directly DNA damage in mitochondria. I was talking before about alterations of mitochondrial metabolism, but affecting probably to DNA transcription and to DNA and to synthesis of proteins, etc., but not modifying the DNA sequence, as far as I know. Okay, of course, we have a very limited amount of data, but the data I am aware of is mainly related to modification in the protein synthesis and DNA transcription and then protein synthesis, translation, but not alterations of the genes, okay, mitochondrial genes or nuclear genes, okay? So I would expect that the alteration may be permanent or not, depending on the ability of the carotid body and other cells to regenerate, okay? But not because of any permanent DNA damage, as far as I know, okay?
Perhaps a more interesting question comes from Benedito Mercado, and he asks, is it possible that some patients with silent hypoxemia actually have already had their sensors in the carotid body cells deactivated before the infection?
This is a very
so that this could actually be the explanation why some people become so much more ill than other people when they're infected.
I think this is a very good question. I didn't go into these details because I was focused on ACE2 expression, okay, the level of receptor ACE2 expression in the carotid body cells. But of course, many of the people who have comorbidities that suffer a very dangerous, complicated COVID disease, and they are hypertensive, they have diabetes, and it is not, as far as I know, there's not a clear demonstration that the carotid body is altered in this condition, but there are some preliminary data suggesting that hypertension and diabetes may produce alterations in the oxygen sensing ability of carotid body cells, and therefore, if this is a very good question, it could be that the problem of some people are related to the comorbidities. And people with these comorbidities, from the very beginning, they already have not a very good ability to detect hypoxia, and therefore they suffer hypoxemia very early in the disease, regardless on whether or not the carotid body cells are infected. Okay, it could be both things happen. The carotid body is not responding well to hypoxemia, and in addition to that, the carotid body cells in some people with high levels of ACE2 expression may be infected by the cells and therefore worsening their oxygen sensing capabilities. Okay, but that's of course it's an expert in the field. Very good question. Okay.
Okay, thank you very much. Alex, there's a question for you. So you put a lot of emphasis on the blood-brain barrier. And it seems, if the person understood you correctly, that this is one of the main problems. Are there any kind of remedies that could specifically help here to repair the blood-brain barrier? And if you are not repairing it, could that again be a reason for a kind of more long-term effect?
Absolutely. This is really very, very nice question. And of course, for the time being, we don't have any curative strategies for repairing blood-brain barrier after it has been compromised. And obviously, it is repaired internally by the cells of the brain. So, for example, the parenchymal part of the blood-brain barrier, they are not only astrocytes, but also microglia. So whenever whatever damage occurs, microglial cells are also participating in repairs. This is all known, but we have absolutely no ways how to influence it. So indeed, that could be a long-term damage, which may participate in neurodegeneration.
Okay, thank you. And a question for Bernard Rossier. He asked, does the evidence you have shown that actually the COVID virus is transmitted via upper airways to the blood first? The actual transmission of virus could then, as regards the lungs, could then be via pulmonary capillaries to the capillary membrane. So this person asks, is then COVID disease not just a respiratory virus, but can affect a lot of organ systems coming from the blood? It's a little bit confusing for me, but I don't know whether you can comment on it nevertheless, Bernard.
Yes, I think it's a relevant question. Obviously, I think this is coronavirus are mainly airway infection first in the early phase, like influenza virus and other, virus of the airways. But of course, then it will go into the circulation, and unlike other coronavirus, it will colonize the endothelial cells. And of course, colonizing the endothelial cells will mean that every organ in the body will be affected, and this causes severity of the disease, a systemic disease, because the lung endothelial cells will be affected, the brain endothelial cells will be affected, the muscle, every organ, and this makes the disease very severe. What I was proposing is really the early phase of the infection, and I think everybody would agree that it's mainly concerning the airways. And of course, the GI. I didn't talk about the GI intestine, but obviously when the mucociliary clearance works fine, it propels the virus into the intestine because you swallow what has been pushed upwards by the larynx and the trachea to your intestine, and then you infect your intestine. And it's well known that some COVID patients have primarily intestinal symptoms, diarrhea, which is maybe also a problem of ENaC-mediated transport, which is compromised by the virus in the colon, and other intestine symptoms. So my proposition is that the main entry is airways, then the propagation into the airways, lower airways, and simultaneously to the intestine.
Thank you very much. Val, there is a kind of more practical question for you by Paul Thomas, who's asked, should people who have just got COVID, or just been diagnosed with COVID, should they use mouthwash to reduce viral load?
We don't have an answer to this yet. I think the clinical studies that are ongoing are testing whether a rinse in somebody who's positive reduces the detectable infective virus in the throat, and if so, how long that effect might last. And the studies I showed, which have come out so far, the numbers of people are too low to really draw any conclusions. They look like they're going in the right direction, but I would say that it's so far anecdotal. And I'm not a public health official, I'm a lipid biochemist, so I couldn't really make recommendations. And right now it's got to be a personal choice for people whether they choose that approach. I hope the clinical studies get enough numbers to report soon so we can at least answer the question as to how effective these formulations are at inactivating virus in the human throat. One issue is that a lot of those two studies used patients who are in hospital already maybe shedding less virus in the throat than patients with early disease who are transmitting virus. So it may be that we need to be looking at people early in the disease to answer that question properly. But data is just not really there that would allow recommendations to be made. But as I said, it is a personal choice, I think, for people to consider if they wish.
Okay, thank you very much. And then there's a kind of very general question, which I'm not sure who would necessarily be best to answer it, but it's from Essentia Ferrini, who said, I have heard that the aim of the coronavirus is not to kill its host, but to reproduce itself. And I saw this teleological argument used to predict the direction of mutation. What is the status of this kind of approach in your scientific research? Does finality still play a role in your studies? So it's a little bit sort of philosophical. I don't know whether somebody is willing. You are sometimes a little bit more philosophical, Alex, than some of us, and whether you have a go at this question, but anybody who can join would be more than welcome.
Well, I don't think anything in the universe has any aim. And of course, viruses don't have any aim. It's all natural selection, and they are supposed to survive somehow. And clearly, unfortunately, coronavirus, COVID-19, doesn't kill the patient too rapidly. If it would, like it was in case of Ebola or whatever MERS, then clearly the whole thing would stop. Now we have a long-lasting disease, and so virus is sitting within this chemical laboratory, and of course it is mutating. And some of these mutations could be bad, some of these mutations could be good, we don't know. So in that sense, we can't predict anything. We can't predict the direction of mutation. So obviously, from the virus perspective, to kill the host is not very clever, because then the virus dies itself. But, you know, virus doesn't have this particular aim, and probably nobody is leading it in its life. So I don't think anything is predictable. It's stochastic, it's natural selection.
Did you want to comment, Giuseppe? Please mute yourself.
Lower corner.
Please unmute.
Yes, sorry, Ole. Just to understanding the evolution of a virus, what is important, not only the mutation of the virus, but also the genetics and genomics of the host, because it is a co-evolution in terms of the future. We need to study of the genetics of the host to understanding what is the future, the prediction of how this virus will be evolved.
Yeah, it's a very valid point, Giuseppe. Thank you.
Another one for you, Alex, that just has come in the gyrus dentatus. There are IL-1 receptors, and IL-1 can be produced in the brain. So cytokine storm may perhaps just activate this internal process in the brain, from a George in Innsbruck.
Absolutely. All cytokines are produced in the brain by the host of different cells, and this is part of normal physiology. Now, what happens in the cytokine storm, indeed, that the BBB got leaky and all these things are coming into the brain, and they overactivate these things, because obviously the brain defensive system, say microglia, are under incredibly tight control, otherwise it will kill the brain. And in this situation, we have a deregulation of the system, and of course, after being gliotic reactive, all these cells would start to produce cytokines inside the brain, which would be a sort of autoimmune inflammation, if you will.
And there's a very specific,
No, just because I noted there is a question on interferon and the BCG. It could be related to interferon. This is important because many people demonstrated that the BCG vaccination could be a protection against coronavirus. But some epidemiological studies recently does not confirm this. Therefore, could be that BCG can increase the levels of interferon, and in particular, interferon gamma. But we need further studies to confirm this.
Okay, Jose?
Yeah, Ole, thank you. I wanted to confirm with Giuseppe that the mutations in the interferon that you found associated to the severe COVID is a loss of function mutation. Okay?
Yes, yes,
okay.
yes.
And you see the mutation or antibodies against interferon.
Exactly,
okay.
exactly.
Okay. Thank you. Very nice work. Okay.
A very simple, specific question to Alex again. Has the SARS-CoV-2 virus actually been measured in the cerebrospinal fluid?
Well, from what I know, and I don't spend all my life in looking into COVID things, I've seen only one paper which found viral particles there. It might be more so, but I simply don't know. The only thing which I probably can say is that if you think about the whole brain, there are viral particles found in postmortem brain, but this is not very, very frequent problem. So it could be even viral encephalitis, like it was described for normal influenza, but this is not very prevalent. That's all I can comment here.
Okay, and then a question for Jose. So it seems that in the glomer cells, calcium is an important link in sensing the oxygen and then giving the activation of secretion from the glomer cells that will then activate the pathways into the brain, stimulating ventilation. So given sort of some of the other aspects of calcium transport and so on, could you speculate a little bit more? I mean, normally we talk about calcium as being important in what we might call stimulus metabolism coupling, so that you need this increasing calcium in the mitochondria in order to activate the Krebs cycle. And do you know anything about whether that specifically is changed in the COVID-19 situation?
Yeah, well, in related to the COVID-19 situations, I'm not aware of anything that related to what you mentioned. We don't have any direct studies on COVID or coronavirus infected cells. Okay, we are looking forward to do some studies in coronavirus infected cells from humanized mouse models. Okay, in which we can remove the carotid body, which is already infected, and we can do some functional experiments, etc. But regarding what you mentioned about a stimulus secretion coupling and stimulus metabolism coupling mediated by calcium, this is very well demonstrated in the carotid body glomus cells. The carotid glomus cells is a sort of presynaptic-like cell. It will release transmitters in response to extracellular calcium influx. This has been very well demonstrated years ago by our group and other groups, but also it has been demonstrated by other groups, by some group in England. For example, Keith Buckler very nicely showed years ago that the mitochondrial function is very much dependent on extracellular calcium influx. Okay, if you remove calcium, the function of mitochondria, the function of the Krebs cycle enzymes, etc., the electron transport chain protein do not work so well. Okay, so calcium, I think the carotid body chemoreceptor cells, which have a very active electron transport chain, mitochondrial electron transport chain, are very good example where calcium influx is activating metabolism, okay, and particularly mitochondrial metabolism. Whether or not it have any relationship with COVID-19 infection or whatever, we don't have data on that. Okay.
Okay, thank you very much. And then another question for Alex. Is there any further research planned, or what could be the plan to investigate further the hypothesis of the expected increase of neurodegenerative diseases post-COVID? Is this further research planned? And what might be the plan? As repeated again, is by Thomas Jaworski.
Well, I mean, of course, I'm not responsible for making any plans. So what I know is that many countries launched specific programs dedicated to studies of COVID. And I would assume that within these programs, obviously, there should be some neuroscience, because clearly neurology of COVID is there. What are the plans? I mean, we have to continue to study the whole things. And of course, in neurodegeneration, there are many, many different factors which have to be studied. And in particular, there is infectious theory of Alzheimer's disease. And it wasn't confirmed yet, but clearly there is something in it. So I think, well, that should be just normal plan of how to study this. That's all I can say.
Okay, okay, thank you. And I think we are sort of approaching the end of this question. There's just another one that just came in. So this is about mitochondria. I think it is for for Jose Lopez Boneo. Could SARS-CoV-2 induced hijacking of the mitochondria activate apoptosis?
Well, this is a good question that I don't have any clear answer. There are very nice publications which appear in Nature a few months ago, looking at the proteome or human cells infected by the virus. And they show there's not only decrease in mitochondrial protein, but some increase in some type of mitochondrial protein. So this hijacking of protein is not only decreasing all the proteins in the mitochondria or in the cell, but also some molecules are activated. So whether or not apoptosis, a release of cytochrome C and apoptosis is enhanced by COVID infection, coronavirus infection, I don't know that. Probably it is published already, but I don't follow closely all the literature on COVID infection. Okay, I was focusing very specifically in carotid body infection. Okay, but it could be possible. Okay, that COVID infection would increase the release of cytochrome C from mitochondria and then induce apoptosis, and this would contribute to the death of the infected cells. Okay, that is a very plausible possibility.
Okay, thank you. And just two questions just came in now. One for Giuseppe Novelli. Given the findings implicating interferon in severe COVID, are you aware of any similar studies in so-called long COVID?
Yes, not yet. But we started with a very large clinical trial using interferon, and we hope to wait. We hope to obtain a very important indication for this.
Okay, thank you. And a question not addressed to anyone specifically, but maybe Bernard could have the first go at it. So the person says leukemia inhibitory factor, LIF, protects the lungs from SARS-induced viral inflammation. Why is it not used?
I don't know.
Does anybody have an answer to that?
No.
I don't know. It could be interesting.
Nobody has.
I know that we use leaf in the lab for other purposes, okay? And leaf is very expensive, okay?
Well, I think there are a number of targets which are tested now, and we don't have yet the results. The clinical data are not yet in. But for instance, the bradykinin storm is one possibility that you should target the bradykinin synthesis and so forth. But the drugs are there, they are already FDA approved and they could be tested, but I'm not aware of any results. The same is for TMPSSR2, that was mentioned by Giuseppe as a susceptibility allele. This is a co-receptor. We do have a drug, Camostat, to block this enzyme. It has been tested in cancers and different diseases, but I don't know if it is now tested in COVID. It could be to prevent the binding to the co-receptor. So I have a question to Giuseppe. I mean, you talk about a lot of susceptibility alleles. What about protective alleles? That would be.
Yes, thank you, Bernard. We're just looking at some genes, and we started with a new project in order to identify resistors people which are exposed strongly to the virus, but are negative to the RT-PCR antigenic test. And now we started to look at the genome of these people in order to identify resistors.
Like for the HIV story. Like.
Okay, I think we have come to the end of the question. I think we have answered the questions that have come in. And unless any of you have any particular sort of final comments, then we are probably approaching the time where we should thank all those who have stayed with us. Just now I see that the number of participants is dropping, so that's probably also an indication. But we have had a good audience that stayed with us until very, very, very recently, almost all of them, which is very pleasing. So I thank all those who have listened to what I think has been some very interesting presentations and a very good discussion. And of course, I thank particularly all our presenters who I think have done an excellent job in actually demonstrating an enormous broad range of research, from a very practical one to very interesting theoretical ones. And no doubt we shall see enormous progress in this area, given that there are masses of people who are working on this huge problem, which probably, as Alex says, will also cast a very long shadow into the coming years. So on behalf of Academia Europaea and also of the journal function, I thank you all very much. And no doubt we may well at some point come back to this theme again, because no doubt a lot of new things will happen, and it may well be in the due course that there will be a need for another webinar on this time, or possibly even, if we are lucky, a real meeting where we can actually be together in a more direct manner. So many thanks, and I therefore close this webinar now.